Hi, everybody. Today is December the 13th, 2024, and this is Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. So today is a special day for us because we have just completed our annual Neuroscience Symposium, and the topic of the symposium was Basal Ganglia Circuitry, and we have assembled this distinguished group of panelists who gave talks in the symposium about various aspects of the basal ganglia circuit structure and its function and its clinical importance and behavioral importance and things. So we have a good representation of the basal ganglia field, and I'm going to just sort of hope that they will just get started talking to each other. They've had no trouble talking to each other up to now over the last 2 days. So I'm going to raise a couple of topics. First, let's get everybody to introduce themselves. Why don't you start with Dr. Mao?
My name is Tianyi Mao. I'm a senior scientist at the Vollum Institute. This is the third time I'm doing this podcast, so good to be back, but scared to be first.
My name is Zayd. I'm a senior investigator at NINDS Intramural Research Program.
That's not your first time on the podcast either.
It is. I've been here before.
Gilad Silberberg, professor from the Karolinska Institute in Stockholm. First time on the podcast. Jim Surmeier, I'm a professor of neuroscience at Northwestern University in Chicago. This is my second podcast.
Yeah, it is.
Josh Goldberg from the TB University. It's also not my first time. I'm looking forward to getting the sticker at the end.
There is a sticker coming for you in the end.
I didn't get a sticker the first time.
Need to work for
That's in the
I'm Chip Gerfen. I'm a senior investigator at the National Institute of Mental Health in Bethesda, Maryland.
And I'm your host, Charlie Wilson. So one of the themes that I noted is that everybody had a different part of it, is this sort of heterogeneity among well-identified things that we used to think of as single things, like a single cell type or a single axonal projection in the basal ganglia. So maybe not everybody shares this prejudice, but it is commonly thought that once we have identified the cell types that make up a brain structure, then we're done, basically. We know the cellular composition of the brain structure. But it turns out, apparently, that we're not finished with that, because as we look across the cell type from cell to cell, or maybe part of that brain structure to the next part, we see variations in the cell type that turn out to be important for the way the connectivity works. And it's frightening, because it may mean that we're going to have to take the brain apart cell by cell and not just brain structure by brain structure. So I'd like to get an idea about the scope of this issue and maybe an example. Somebody want to raise an example that you offered us during the talk? Zayd, do you want to talk about dopamine neurons?
Sure, since you mentioned me. Yeah, so the dopaminergic cells, one early on idea was that they might be basically uniform and doing exactly the same thing, but maybe participating in different brain circuits, but doing exactly the same thing. As it became sort of clear that the dopaminergic cells were, not necessarily all participating in reward or circuits, and it just, started to become more clear that maybe the function of the cell and the circuit that it's involved in may be performing different tasks there. And so you have to start to accept the idea that maybe the cells themselves might be different in the circuits, right? And so, there could be a lot of ways to get a heterogeneous cell type and circuit to contribute to different behaviors. And one of them is to just have different functional cell types. One of them is just to have, so it looks like the dopaminergic neurons, there are different functional cell types that have different intrinsic properties. And different connectivity and
We're having an Amber.
different Amber Alert responses.
Is that one of us? Okay, good. I'm glad the emergency is over.
I'm sorry, that was mine. I know, Amber, there.
Yeah. So dopamine is a great example because dopamine has sort of worked itself into popular culture. People say, why do you do that? Well, I get a bit of dopamine out of that. Or why do people do what they do for the dopamine? And the notion is that somehow dopamine is a thing in the brain that has one function.
Right.
And so the finding that some dopamine neurons behaviorally are active in completely different circumstances than others, and then the other idea that causes dopamine to get inserted into popular thinking is that somehow dopamine drives learning. It's the reinforcement signal that drives my learning. And so, do all dopamine neurons, are they all reinforcement signals that drive learning? It seems like maybe not.
I don't know. I mean, it enables movement, right? And I guess that's not learning, right? It has to be around, and so it enables movement. And so I guess in that way, it's not a character signal, but it's a tonic signal that enables the function of the DLS, for example. But in many circuits, it is driving learning, right? It's important for the imprinting of a event.
But those are not mutually exclusive, right? It's possible for dopamine to control the vigor, for example, or motivation to move, and as well as to have longer-term consequences as to what your actions may be in the future through different mechanisms. And the moment-to-moment changes in excitability are well documented that dopamine inserts, and those seem to be fairly similar across different basal ganglia regions. The plasticity mechanisms, it's not quite as clear that they're similar, but the data that we have suggests that at least in regions where you might think dopamine is related to movement vigor and maybe not so much to reinforcement learning, the plasticity mechanisms are very similar. That is, dopamine can enhance long-term potentiation in some cell types and long-term depression in others. So I don't think they're mutually exclusive.
Another thing we talked about was the difference in the striatum. Dopamine is acting in striatum in the situations that we've been talking about. The differences in striatum of function. So there's like the auditory part of the striatum. We talked about auditory. There's a visual part of the striatum. There's some overlap. There's like uncertainty about how much overlap there is. But dopamine axons don't go everywhere in the striatum. They're big. True, but they go to particular places. So could we expect to find dopamine neurons that are exclusively involved in auditory learning tasks and others for visual learning tasks? Is there any reason to think that exists? That's the kind of thing one could look for.
That's a good question. I guess many of them do respond. So the neurons that respond to salience are the ones that are in the lateral sort of substantia nigra and the ones that are in the SNL. But many dopamine neurons do respond to cues, right? And that's like a sensory stimulus. And I'm trying to think of a neuron that, go on.
Well, so I'll chime in here.
Yeah.
And so it is very clear that some dopaminergic neurons will respond to reward, for example, and others will respond to punishment, that is, to aversive influence. And that those two dopaminergic neurons must have very different functions. I mean, what's driving dopamine release and spiking activity is very different. They target slightly different regions of the brain that are involved in either avoiding punishing situations, or those that are involved in getting us back to rewarding situations. So I think that, there clearly is heterogeneity in the regulation of those different dopaminergic neurons, and then their targets involved in performing different functions. So I think there's good reason to think that there's heterogeneity in the functional properties of different dopaminergic neurons.
Yeah. Also, but many of them do respond. I was more thinking, you're totally right. I agree with that, Jim. It's totally right. But many of them broadly sort of respond to sensory stimuli. So that's what I was saying. And I was trying to think of it, are there dopaminergic cells that I know of that don't really respond strongly to sensory stimuli, although they do respond to, rewards. Some of them don't respond to rewards. I can't think of any that don't really respond to sensory, like auditory.
Yeah,
cue
so
elements.
how do those neurons distinguish between sensory events that are aversive and those that are rewarding? I mean, so we use this term salience
Yeah,
to sort of be a grab bag
right.
for things that could fall into all of those categories, right?
Mm-hmm.
In fact, it's a word ordinary people don't, like me, don't know it, even want to know what it means.
Salience.
Yeah.
Yeah, no, it's hard to
That
determine.
category, yeah. What's salience?
Yeah, what salience is. It's
Right.
just something happened, right? But I mean, a new reward is something happening, and I just showed in some cases, some neurons don't respond to a new
Yeah.
reward.
A harsh
So-
punishment is salient, but I sure wouldn't do anything to get that.
Right. So, I mean, one of the interesting things is that cholinergic neurons in the striatum, which are playing this game with dopaminergic axons in the striatum and regulating the circuitry, are very well known to respond to salient events independently of the valuation of the stimulus. Right. Something like a clap or something like that, where you don't know whether it's going to be a good thing or a bad thing, but something that demands your attention. And so it'd be interesting to know whether the dopaminergic neurons, whether the potential, what's the word I'm looking for, value of the stimulus, or I'm blocking right now on the term that's used. Another term that's
Just-
only used
just
by
as well.
specialists like
Just as well.
salience.
I probably wouldn't know what it means.
How that
Valence?
sorts out. Huh? Valence, right.
Yeah, valence.
Valence, right.
That has to do with how many electrons there are.
So exactly. I like the word electrons.
Another thing I think that we should I think about is that people associate dopamine with positive effects, positive rewards, whereas the negative effects of dopamine are, I think, just as important. And I often have heard people talking about, they're really surprised because there was rewarding through the indirect pathway was involved with learning, and dopamine was involved in reward mechanisms with the indirect with suppressing possibly certain movements. Well, that's an effect of dopamine that might be considered opposite of something that would be considered positive reward. And so that's another aspect of dopamine that is often not, we don't think about it. And go ahead.
No, I was just going to say that the one of the things about dopamine is there's a constant level of dopamine in most target structures, and the conventional view is that it's bidirectionally regulated, that it goes up with positive reward and down with negative outcomes. But as you pointed out, there are dopaminergic neurons that you've seen in lateralis, and we see in the paranigral region, where the dopaminergic neurons are actually excited by aversive input, and exclusively aversive input. So it's another variation on that complexity that we haven't appreciated to this point in time.
And also when we're thinking about the action that the dopamine is released, and it does impact the T2 through the possibly suppress the activity somewhat. That's something that is actually important for a positive outcome.
So if I may summarize, it seems like there's some dopamine neurons that fire when good things happen, some fire when there bad things happen, some fire when there's just something important that isn't neither good nor bad happens.
Mm-hmm.
And those are different neurons located in different parts of the dopamine system and projecting to different places in the brain. What makes those all one nucleus?
Well, I was going to get back to showing that this idea that dopamine, we're up thinking of dopamine neurons are how we could see it, because we could identify them as expressing ETH or tyrosine oxalase, or the methods, the thing that actually I started, and you maybe you did as well in the field. When the fantastic thing about dopamine in terms of brain science is that it was the first chemical that you could actually visualize neurons that express that, because the Swedish investigators figured out that they could use this chemical reaction and cause dopamine to be able to condense, and it became fluorescent. And that was an incredibly magical time, because all of a sudden, you could see where dopamine was in the brain, and dopamine was in certain neurons in this part of the brain. That's what led to the identification that Parkinson's disease was the result of dopamine being lost, in that part of the brain.
It was huge. We take it for granted now because we can do that one day.
And we don't think about it, because that was the beginning. It was the first time that neurons could be identified as different because of the chemical, the neurotransmitter that they were using, because of the catechol fluorescence method. And then, the next step was antibodies or other methods to be able to visualize neurotransmitters or chemicals that identify neurons to be very specific. And still, what was the dopamine neurons? We saw that they were mapped out, and we saw that there were different parts of the brain, there were different nuclei, and so we got names like A1, A2, A10 with PTA and all of these things. So that was the first demonstration of that. And then we got to thinking, okay, well, the A9 is compacta, but then it turned out that, oh no, when you look closely, you could see that some of the compacta neurons had dendrites that were spread in the plane of the dopamine neurons, and others had dendrites that were spread down. So this terminology came out that there's a dorsal and ventral tier, and then tracing methods show that they actually had distinct projections. The dorsal and ventral tier connected differently, and it came along with other neurochemicals. How bendin was shown in the dorsal tier, how ALDH was shown in the ventral tier.
So what is it in? I mean, what's the...
And so now...
Perhaps
now
we should give up on this idea.
Now we're in the point of, like, people can take with the single cell nuclear sequencing, and identify 50 different types of dopamine, not 50, but 25 different types of.
How many do you think?
It's isn't it 40? Or some papers had 40, some papers had 30 something types.
Yeah, and so this is how this.
Mouse,
Some had 25.
20,000 of them altogether. So if there's 40,
so maybe there are 20,000 exactly different cell types. I mean, I wonder about this sort of. There are always splitters and lumpers, right? And it's never clear to me how much of a difference is biologically meaningful in gene expression, for example.
Mm-hmm.
The other thing is that it's become clear that, particularly in disease states, the phenotype of dopaminergic neurons is plastic. That is, they can change their stripes, right, to become leopards or something, because we've seen an enormous amount of plasticity in response to challenges of various some of these sorts. And I just wonder, even in a healthy brain, how much plasticity there is in this dopaminergic neurons. I mean, as well as any other neuron in the brain.
This gets back to a core concept. What is the cell type? Right? In this talk, 2 days, we talked about specific subregion location, transcription factor, dendritic morphology, their projection targets. But all this grouping, not necessarily, the same gene or two genes give you two distinct morphology and what they connected to. And they respond to functionally distinctively. And then reverse problems that when you see some cells respond to positive, some dopamine neurons respond to negative, you don't necessarily have a way to go back to the same cell. So Charlie said, these are different cells, but how do we know they actually are different cells instead of the same cell that play a different role across animals? But I view this as the beauty of the basal ganglia system, is that where you look at the D1, D2 neurons, where morphology, transmitter, their input-output, genetic targeting, everything sort of beautifully in there and functionally. That's why I think this field has been seen growing knowledge that because the cell type segregation, there is truly functional, but you have access to them. I don't know how far we go with dopamine cell. Like there are beautiful work from like say Raj's lab allow us to access them. But how we would next step going to have a true understanding of cell type that you go through all these 5 different categories of the cell property to say this is a cell type instead of this is a cell that function different from your neighbors.
We don't want to deconstruct the idea of cell types completely, because everything we do is based on the idea that in the striatum we can identify 2 kinds of spiny projection neurons, and that they have different morphology of their axon, different projections, and different gene expression and different function.
So I think it's also a reflection of the scientific inquiry. I mean, we usually look for general principles, and maybe that's when we're reductionists. We start with something simple, and then as time progresses, we talk about lumpers versus splitters. So first we focus on what's the common thing, and then we start looking at the heterogeneity. But I think also Jim mentioned an important point is that all the neurons we study, while they might have various heterogeneity in various dimensions, they also have this homeostatic ability to sort of correct it. If there's something different about them, there's this buffer zone. So maybe heterogeneity isn't that important because they can move around in boundary space, and so it'll be there because nature is stochastic, but the system has feedback in order to keep it in some sort of functional range. So maybe heterogeneity is not such a big
Not so
problem.
scary.
It's just scary.
Well, don't you guys think that the connectivity is like kind of a defining feature of a cell type?
So here's an example from Charlie. Is that one of the things about the striatum is spiny projection neurons. 95% of the neurons in the striatum are spiny projection, 90% of whatever. So I used to always give talks that say 95% of the neurons in the striatum are spiny projection neurons. Morphologically, they all look alike, but half of them project an axon that only goes to the globus pallidus, and the other half is an axon that goes to the substantia nigra and directly to the output. So, and then that was okay. There's 2 types, except that Charlie pointed out in your initial thing said, well, there's a collateral into the globus pallidus, so that goes into the indirect pathway. And I said, yeah, but it's a little collateral. Oh yeah, it's not that, but it's back. And then there's been studies in not pathologic, but there are conditions where you can actually get that axon that goes into the globus pallidus from those direct pathway neurons. It expands to the point where it's almost as great as the one. And so that's an example where the connectivity actually can change plastic.
Yeah, it's also true in models of Parkinson's disease. You do partial lesions, and one of the things that was described 30 years ago was sprouting of axons to sort of invade the regions that have been de-innervated. So, you wonder, even in non-pathological situations, how much of that's happening. We don't really know.
So the cell types and axonal projections and all that stuff we study could be dynamic things that are being created in real time, even in adults, so that what we're looking at isn't a permanent, forever difference between
Jim's
cells.
example was showing the spine, the spiny neurons can alter in terms of that they can clean, they can expand, whatever in different conditions. So that's another thing.
So in that case, understanding cell type would mean understanding the forces that create cell types and that sustain them and keep them from all just.
But there are some rules, plasticity can react in a homeostatic way, but it cannot usually turn the circuit upside down. You won't start getting SDMs projecting back to the cortex. This directionality will remain. So there is homeostatic, but it has its envelope or limitations. And regarding the point that you mentioned about the collateral, it's actually very important to know what they target in the GPE. So the DNSNs or DSPNs, they will specifically target archipelagal cells in the GP and not the prototypical, the majority. Yeah. So it's not just the fact that you have an axon there. The axons are often very target selective. Yeah.
There's a question from the audience. Dr. Michela. Well, that's what we're trying to figure out. Alfonso is asking, how do we define cell types? And, we've defined them just sort of ad hoc, historically. We've said, and different people have different criteria. Some physiologists describe cell types according to their responses. Here's a rapidly adapting cell and a slowly adapting cell. And I declare those to be different cell types, which they wouldn't be by the criteria that we've been using. We would have to see that they express different genetic constellation. We would have to see different morphology. We'd have to see something other than just
I did my PhD in the lab of Henry Markham when he was very interested in diversity of interneurons in the cortex. And it was a time that we could explore this diversity at the electrophysiological level, then the morphology, and then the target, the projection. We came out with 40, 50 different cell types, and most of the community was quite angry, kind of shooting the messenger for that. Okay, I'm showing you the diversity. I'm not claiming that each cell type, we know their exact function, but at least we have to acknowledge that it's there. What to do with it is something else. But splitters exist. They don't invent new cell types.
Yeah, but test our desire just to have a small number of things to think about at a time.
But the problem is conceptual, in my view. Is there a continuum, or are there discrete groups, right, where there really are significant gaps between the distribution of properties in some sort of multi-dimensional space? Right? I don't think, and I think the way we think about the function of cells is actually impacted on whether or not there are discrete categories or whether it's a continuum, right? And how the brain is working. So I think it still is an important question to kick around, right? Whether the discrete cell types or whether there's a continuum of properties, because I think it has implications.
Well, I look at it also that it's part of the job. It's part of the fun job. So I think they just have to, or have chosen the career we have, is that we can take this diversity, and the fun part of it are like is to try to figure out, extract from that, what are the basic underlying principles of which groups, what groups do we extract from this diversity, and then those groups that create the model of how we use the function.
As our collection, we figure out different ways of categorizing them and putting them in different boxes. Like, how do you organize your reprints? Some people do it by first author name, last author name, topic, journal. Every different way of organizing it is possible.
But the implicit assumption is that any categorization like that is going to be important to the function of the brain. But that's an assumption.
Well,
right?
Some are and some aren't.
I mean,
and there's a lot of people have ideas. All of us have ideas. A lot of the ideas we have to share with other people because it turns out they're not very good. And other ones we end up, being important. We share with people that anyway. That's exactly. So we can come up with a lot of ideas. That's the fun of it. It's like, okay, which are the very important to how the brain functions. They'll say, no, I disagree with that. No, I think that's part of the argument.
So Charlie, you mentioned sort of the public opinion about dopamine, and I'm sort of thinking, what's the public opinion about neuroscience at large? And this year, the Nobel Prize went to two physicists who I think also pissed off the physics community, but they basically described two very, very abstract principles that neurons could do, and Karolinska's imagination, Hinton and Hopfield. And there, all of this stuff about heterogeneity and the differences between neuron types really is important. The principles people like they're captured by very simple representations of neurons.
So the inference from their work is it's not important.
Right.
Right.
And in that case,
Dendrites are not important.
Yeah.
Dendrites are not
There,
important.
we've definitely hit a nerve out in the audience. So, the camera went dark.
We're still seeing it here.
It's okay.
Anyway, yeah, so maybe it's not important at all. We can treat neurons as uniform point functions.
So can I bring up another topic? Because there was another theme that I really heard during the symposium that sort of caught my imagination. And that is, it's another old idea, but it's an idea that really has become real, I think, in basal ganglia. And that was that there's sort of two time frames of neuron interaction. There's fast synaptic transmission, and the people who are interested in that track out pathways that really care about synapses. If these two axons are near each other, but they don't make a synapse, and they're not going to communicate. All of that was an established way of thinking about the brain. And when I started, that was the correct way to think about the brain. And John Eccles, kind of way of thinking about the brain. But at the same time, there was this knowledge that there were these modulators. That at the beginning, there were just acetylcholine, I think, might have been the first one, and dopamine, and then peptide transmitters. And those seem to not really require synaptic connections everywhere, and they are acting on a much slower time scale, and they work spatially because they depend on the diffusion of molecules, not the punctate exact location at synapses. So things that are just near each other in space, even if they have no special relationship to each other, will interact with each other in that way. And so now I think most neuroscientists think both of these things happen at the same time in the tissue as they're studying them. But people pick one. They'll study one of these or they'll study the other. But at some point they have to touch each other and interact, the slow time scale and the fast time scale. And there was a lot of that.
So like Zayd's example this morning, that it's not only temporal, but it's also spatial. It's this idea that acetylcholine is going to make the dopamine axon fire. It can't be at a point. It has to sort of do it to a whole piece, a big piece of it. So I think it's a spatial-temporal question. Scale.
Stand correct.
No, I think we touched upon it when we talked about the different neurotransmitters, that they have the receptors that are fast and slow. So when there's a release of acetylcholine, it will trigger slow processes and fast processes with the G-protein coupled receptors and to the ionic receptors. Same is for the glutamate and for the GABA with GABA A, GABA B. Turns out that also with dopamine, even if there is no dopamine fast receptor, there is a release of dopamine with GABA glutamate. So even when there is activation of dopamine cells, you will have these quick processes and the slow processes at the same time. So almost all of them, all of these neurotransmitters that we work with will have a slow and fast.
So this tells me how they're separate. The fast network can create slow things.
Yes, and vice versa.
So the other side is how that slow network alters the fast things, or even those things on its own. So
right. I mean, in the striatum, it's a great example. I mean, dopamine and acetylcholine are controlling the induction of plasticity. There is the strength of glutamatergic synapses that rely upon inotropic receptors. They also modulate GABA receptors. So the other sort of major fast neurotransmitter. And in that particular case, it's clearly associated with learning. But it's not so clear in all the other cases what the neuromodulators are doing to the computations that the brain is performing on a fast time scale. And it's clearly affecting both receptors, but also all the ion channels that control the integrated mechanisms in whatever computation a dendrite or a cell body is performing. I think it can, whether it changes it in a fundamental way or on some sort of sliding scale is not so clear. But many things in our lives are slow. already. And so I think I don't exactly have a good conceptual framework for integrating the two things. I think we've identified a really important problem, but we seek evidence for interaction. The question is exactly what does it mean? In neurological diseases like Parkinson's disease, the trigger clearly is loss of the slow transmitter, not a fast transmitter. So, I mean, it's not universally the case, but in Parkinson's disease, it's true.
I have a question about movement vigor, though, right? That's not necessarily like learning, for example, but if you increase dopamine release, you can get the animal right there when you do it to generate more vigorous movements. And that happens through these slow receptors, right?
Yes. But through not through long-term changes, but changes in the moment-to-moment function of ion channels that govern synaptic integration and spike generation.
Right. Mm-hmm.
And so the cell will forget.
Mm-hmm.
Right? You need to have forgetting, right? You want to increase the vigor now, but maybe a few minutes later you want to slow down and stuff. And so that it has to be a fairly rapid time constant of decay for those effects. But yeah, this is one of the things that I think is really important for us to get our heads around, is that these neuromodulators, beyond inotropic, very fast synaptic transmission and slow stuff. Even on the slow events, there are different time courses. There's the seconds sort of time course, and then there's minutes and hours for days time courses that are involved.
There's also variation in the spatial resolution, because some of them diffuse very rapidly and easily, and some of them diffuse much less. So I'm imagining, and this is kind of fanciful maybe, but I'm imagining that the volume of a brain region and this sort of three-dimensional image of what each of the modulators is doing. Like maybe there's a blob here that's got a lot of acetylcholine and one here that has less. Now, whatever is happening in that space, which is not necessarily defined by location, because the Schneider and fast stuff doesn't necessarily have a precise topography in space. But whatever function is mapped to that blob is going to be affected by that, and the one that's mapped into that blob will be affected by that. And that, how blurry is that image? Is it super blurry, or are they small? Do they move really rapidly, or do they stay constant for a long time?
And it varies from tissue to tissue, right? So in the striatum, there are high levels of data expression which keep the dopaminergic signal spatially and temporally restricted, whereas in other parts of the basal ganglia where there's dopamine release, there's very little data. So the image gets blurrier and blurrier, right? And so the function must be different in those two regions.
But even at the level of like where you have a release site where dopamine is released, is it really well known where the DAT transporters, where the DAT is at the subcellular level? That's and also acetylcholinesterase, for example, is that actually known at the subcellular level? Where are there splotches of it in places? And that I think that was
one of the
really
early descriptions of patch and matrix, right? It was based upon cholinesterase.
Right.
We forget that.
Right.
So some of us have forgotten that. Right.
So
but it goes to your point.
No, but I mean,
But that's
a macroscopic
was the thing where if you didn't stain a long time, there would be patches that change. But if you let the reaction go 5 minutes longer, they got closed in. And so
it means there was variations in how much cholinesterase
in terms of what was going on between these two. Right?
Mm-hmm.
But then it turned out later, it was to show that in fact the neurons in the patches were distinctly neurons in the surrounding matrix, and the dendrites of the patch neurons didn't go outside the boundaries. And how were the patch matrix defined? They were defined by the other chemical markers, the open subcellular axis, because you could actually look at that. And then doping input came, like that.
That's a good point because mu opiate receptors respond to one of these modulatory.
Right, and so it's a way that you can actually restrict how a modulator is only affecting this part of the circuit. I also wanted to just raise this thing about I always talk softly would say that spinal neurons don't do anything that the cortex doesn't tell them. So the cortex, I always think that what's going on fast is occurring in the cortex. That information being funneled through the striatum. Things are happening to it fast and slowly and all of that. But with nailing the lungs, treatment for Parkinson's disease by lesion, the Globus pallidus, I was like, wow, he's like eliminating the output of the basal ganglia. And it's a treatment that people were fine. They could be fine. The one thing that I would ask him, and he had patients who play the piano. It's like, well, that's a really incredibly skilled action that they could do without their basal ganglia considered to be functioning. And like, what could they learn something new? And the answer was not so well. So it might be that what is the basal doing? It's important to learn it. But you can do without if you don't have to learn anything new. You actually get the basal ganglia perhaps. And I found it kind of striking. Like we work on the basal ganglia, it's doing this stuff will actually.
So whatever the basal ganglia is doing is important. The structure is 500 million years old.
I know, it's incredibly important. Yeah.
And so, but if you think about it in terms of rapid, goal-directed action, particularly in like prey-predator situations, where you don't have to really think about what the next movement is, it's based upon past experience or habit, then it begins to make some sense. And I think professional basketball players or football players, sports players, need an intact basal ganglia to function at the level they do. Right? But that may have been important to survival when we were living on the savannah, but not so much anymore, unless you're in a demanding, profession.
Except that one of the things that's been illustrated is that professional athletes are a good example. They're really good because they practice and they do things over and over thousands of times and it's ingrained. So actually, a lot of them are not thinking that maybe they don't need the basal ganglia. They're thinking that Michael Jordan would go up and then switch hands and really like mid-flight and don't let the dead after the fire having an attack basal ganglia or was it because the circuit.
I think that practice is a reflection of the functioning of the basal ganglia.
Yeah, so it's important. It's important 1,000 times to practice it, but then it's actually done.
But isn't that supposed to reside in the basal ganglia? That's a function of the hardwired BLS circuitry.
Yeah.
Is what my understanding is. Yeah.
So that's the hypothesis. But no one's actually done the experiment, right? Mainly used to say, if I had a magic spoon to scoop out the basal ganglia, my patients would be fine, which is a sort of variant of what you say. Yeah. No one's done that magic spoon experiment to see whether the professional athletes function poorly. My guess is they would.
Goldberg did it with the rats. Using cortex, they did an experiment and showed that actually the cortex was important for the learning, but not the execution.
The other way around.
Yeah, the other way around.
I hope Michael Jordan now listening to this podcast.
But I wonder in this case, it is very clear that there are other motor control systems that are perfectly capable of, this again goes back to Nealon's example. Patients can move without the basal ganglia. There's no question about that. It has to do with the speed of the movement in response to environmental events or cues or stimuli or something. I wonder in these experiments, was the speed of the movement without the striatum as good as it was with it?
No, I think the striatum was shown to be more influential on the execution than the cortex.
Oh,
So
I'm
that's
sorry.
I thought there was something recent that it was the other way
No, I think it's the Vretsky papers that I wrote. The training was more important in both the learning and the execution. While the cortex was you could get rid of it and just want to learn the execution. And then I wrote these studies of decorticated cats. They could move around and do some functions, but they're not normal cats.
But they can't learn cats to say.
No, and their interaction with other cats, they were very aggressive. They were not normal.
Oh.
The fact that they could move is something that
This goes back in part.
Dr. Kita points out that this is a species-specific game. This is a worry for us because we mostly work on mice, and we don't want to just explain mice.
Right. I mean, one of the things about that, and I grapple with this too, because Parkinson's disease, for example, is not something you see in even non-human primates. It's something that's exclusively human, right? And so why is that? I mean, it could be lifespan, but it could also be that the organization of the circuitry involved in Parkinson's disease has changed somehow in humans, particularly with telencephalization.
Maybe that's
So you say that humans have more plasticity than growth.
I would posit a very too. Just humans might be combinatorial explosion as far as the cortex is concerned, in that basic English metabolically can keep up with that way. There's some transition between those experiences. Just the number of cortical modules it has to deal with now is so large that it is a finite.
who would like to repeat that question?
Well, I guess the question is how the expansion of the cortex in humans. It has
the experience of the cortex in humans is so large, maybe that explains why to metabolically the basic movement.
right, particularly the striatum, because the striatum has grown in humans has grown almost as much as the cortex, right? It's most of the innervation of the, particularly the more rostral parts of the striatum, are innervated by the cortex. It's pulled, the expansion of the cortex has pulled the striatum with it. But there's not been as great an expansion in the other parts of the basal ganglia. So yeah, I mean, this is one of the reasons why, again, I think that PD is a human disease, because along with the expansion of the striatum has come the expansion of those axonal arbors and the computational burden placed upon the smaller group of dopaminergic neurons. Yes, yeah.
The one structure of the basic ganglia that has expanded is the subthalamic nucleus.
Oh, really?
Because if you look at the subthalamic nucleus in species we work on, rats and mice, it's really tiny little thing. And the human, the primates, it's huge. So it's an example. And I'd like to see what the numbers are between the striatum is expanded as much in the cortex. Because the cortex, because of its, it's the area of the cortex, you count up is it by area or by volume? And
Yeah,
there's an interesting thing to know about whether or not, because I don't know that the striatum is expanded as much as the.
I don't think it's expanded as much, but so the allometric relationships between brain volume and region size are, as I understand it, and it's not work that I've done, it's only thing that I've read, have not changed so much for mesencephalon and for some of the diencephalon as much as they have for Cholin-Saf1, and the striatum has maybe not kept pace with the cortex, but it's expanded more than the allometric relationships would have predicted it would grow. So.
Well, maybe this is a good place for us to stop. So thank you very much, everybody, for participating.
Thank you, Charlie.
Thank you.
Thank you.
And it's been Neuroscientists Talk Shop.